What attracted you to that field of study?
I wanted to be a vet when I was little, and I thought I still wanted to be once I came here. And I love biology still, but I realized that larger organisms just weren't my thing. I think that they smell bad, and they're a lot to handle. And then I discovered that I was kind of okay at bioinformatics. So then I started to explore the more cellular aspect of biology. And so now I love microbes.
Nice. Nice.
The Bennett lab typically researches Streptomyces, which is a genus of gram-positive soil-dwelling bacterium. They're actually responsible for that smell in the air when you smell like the soil when it rains or when it's about to rain. That's the work of Streptomyces. They're also responsible for the production of over 2/3 of our commercially available antibiotics. So they're pretty important in the forefront of medicine technology.
The ultimate goal would be to identify new genes involved in the cell division process. These could potentially become targets for antibiotics. So Streptomyces that we work with in our lab, it does not cause disease, at least not human disease. And so these cell division genes, though, are usually highly conserved. And so they're found in all kinds of bacteria. Ones that people might be familiar with include E. coli, common cause of food poisoning, and all kinds of other pathogens like salmonella, etc. If we can find new ways to develop antibiotics to target other processes.
I did Dr. Bennett's bioinformatics class, and I learned that a lot of the stuff we were working on were techniques that were useful in her specific lab. So I just asked to work in it and try it out, and I really liked it. For my project, I'm working on specifically Streptomyces silicolor. I took on this project that was previously worked on by Alexandra Sherman. I think she graduated last year. So basically, during her portion of the project, she was able to spontaneously generate 3 mutations in Streptomyces silicolor. So when these genes are silenced in a lot of other bacteria genuses, or actually in all common bacteria genuses, it just completely stops them from replicating and living. Whereas in Streptomyces, if you cancel it, it'll cause some deformities, but they can continue to live, which is really interesting. My portion of this project right now is to construct new strains of these mutations. So I'm constructing a strain with an overexpression of the gene that we're studying. I'm also constructing a strain of it with a deletion of that gene to see what happens. And we don't know if this is going to be a gain of function or a loss of function once we do these things. So it's really exciting.
Lexi Sherman was a TA in one of those courses, and she went on and did an honor, or actually a distinction thesis. And then Feng-Thea is even going further so that we can actually get a publication out of this. And so both Lexi and Feng-Thea would be co-authors as undergraduate students on an international article. There's a lot of opportunity from having a publication as an undergraduate because it's so rare. And so that's really seen as something significant. But yeah, there's not many that get to do that.
I haven't quite processed being a co-author on this, even though whenever I've presented at national conferences, I am technically one. I think it's really exciting because I never would have imagined being able to be a part of her lab. I never even saw myself going down this career path. So, I don't know. It's exciting. It's new. After Otterbein, I plan on attending graduate school and perhaps earning my PhD to become qualified enough to do some sort of scientific research. I do want to work in industry rather than academia. I really like the work that Jackson Labs does, or Jax Labs. I don't know if I want to work for them, but I do like to dream sometimes. I think it's very difficult to decide what my exact career will be in the future regarding microbiology, just because there are a very, very large amount of them. There is a wide variety of microbes, and it's very difficult to decide what my niche is with all of those little organisms out there. Right now I'm working with E. coli and streptomyces, but I don't know if that's what I love doing right now. I imagine in the future that maybe I'd be doing some sort of work with viruses, although I have not had experience with them yet. I think that any type of pathogenic research is really interesting, especially because viruses are, what are they? Especially because viruses are built different, to say at the very least. So I think that'll be really interesting.
There they go.